Hi, my name is Andrew Aby, and I'll be doing my biology bonus videos. So I'll do them on for cellular respiration. So let's talk about the chemical equation for cellular respiration. It goes like this: glucose plus 6 oxygen makes 6 waters, 6 carbon dioxides, and 36 to 38 ATP. So let's break down the whole overall process of cellular respiration. So cellular respiration can be defined as the process by which living organisms break down glucose into energy they can use. They break down glucose using oxygen into water, carbon dioxide, and energy they can use, which is ATP. So the four components of cellular respiration are glycolysis, pyruvate oxidation, Krebs cycle, and ETC, which stands for electron transport chain. So the glycolysis, what that consists of, is taking a glucose and breaking it down into 2-pyruvate. But before we get into glycolysis, let's talk about the structure of the mitochondria, because that is involved in cellular respiration. Mitochondria is the site of cellular respiration in all organisms. So mitochondria is a double membrane organelle. It has the inner membrane and the outer membrane. Inside the inner membrane actually consists of the matrix. And the inner membrane is also involved, contains the cristae, which is where the ETC is located. And in terms of location, the pyruvate oxidation and Krebs cycle occur in the matrix, while the glycolysis occurs in the cytosol, which is outside the mitochondria. So let's back up and go into glycolysis, the first part of cellular respiration. This involves breaking down glucose into 2-pyruvate. 2 ATP is initially used, and you get 4 ATP, which makes 2 net ATP produced. And you also get 2 NADH formed, which are carrier molecules which will be used in the ETC later. NAD plus takes 2 NAD plus takes 2 electrons from glucose, actually oxidizes it, so that it loses electrons, and so that it can become 2-NADH. And then after that, ATP produced in glycolysis is actually produced through substrate level phosphorylation, which is when you take an phosphate from an organic compound and transfer it to ADP to produce ATP. So glycolysis overview. It does not require oxygen. 2 ATP is produced, 2 NADH is produced, and you're left with 2-pyruvate, where most of the chemical energy is still stored. And then we go to pyruvate oxidation, which is in the matrix. Pyruvate oxidation consists of 2 CO2 from 1 pyruvate. A CO2 is cleaved, and an NADPH is produced. And as well, coenzyme A attaches to produce acetyl-CoA, which is the end product of pyruvate oxidation. And that happens twice because you have 2-pyruvate, if you remember. And since we are done with pyruvate oxidation, which happened in the matrix, we go into the Krebs cycle, where most of the ATP is produced. And this is produced through oxidative phosphorylation. So before we get to that, let's talk about the cycle for Krebs cycle. So for one acetyl-CoA that enters, it actually does it twice, since there's 2 acetyl-CoAs. So for every cycle, you get 3 NADH, 1 FADH2, another carrier molecule that transports electrons. And then you also get 1 ATP produced through substrate level phosphorylation. And you remove 2 carbon dioxide. So in total, for both cycles, you get 6 NADH, 2 FADH2, 2 ATPs, and 4 carbon dioxides are removed. So afterwards, after Krebs cycle, we're in the matrix, right? We go into the ETC, electron transport chain, located in the cristae of the inner membrane. The ETC is actually the first time you produce ATP through oxidative phosphorylation. So ETC, that's where the NADH and the FADH2 drop off their electrons. So ETC stands for electron transport chain. So basically what happens is that the electrons get transferred through members, and then each electron ETC members alternatively gets reduced and oxidized, meaning they lose electrons and they accept electrons. So this flow of electrons actually releases energy, which is used to pump hydrogen into the intermembrane space, which is between the inner membrane and the outer membrane. Okay, so once we have hydrogen in the intermembrane space, we create a hydrogen ion gradient. This causes the hydrogens in the intermembrane space actually flow through the ATP synthase, which is found in the inner membrane, flow inwards, flow into the matrix. Every time a hydrogen flows through the ATP synthase, you get ATP. That's how oxidative phosphorylation works. So once you have got all the hydrogen, once you've built up the hydrogen ion gradient, it goes through the ATP synthase. You create ATP. This is where about 34 ATPs are produced. Like, the substrate level phosphorylation barely produced any. We get 34 from oxidative phosphorylation alone. Lastly, the last electron transport acceptor is oxygen, which binds with electrons and hydrogen to form water. That's where you get water in the reaction. And that's basically it. That also sums up chemiosmosis, which is where the hydrogen ions flow through the ATP synthase to produce ATP. So what we get, and lastly, really quick summary of fermentation. That's what happens when you don't have oxygen. That's anaerobic respiration. There are two types of fermentation: lactic acid fermentation and alcohol fermentation. Lactic acid fermentation, pyruvate is converted into lactic acid, and you get NADH being recycled into NAD+. So that glycolysis can continue again and again without oxygen. Another fermentation is what yeast use. That's alcohol fermentation, which converts pyruvate into alcohol and carbon dioxide, which recycles NADH back into NAD+, so that glycolysis continue. So the main summary of fermentation: that's what happens when you don't have oxygen, and you only get 2 ATP from the process because you're only going through glycolysis. But the way you recycle NADH is converting pyruvate into another product. And thank you. That was cellular respiration.